Hello everyone, how was your day? I hope you still have the excitement in you to learn more on biology. Before we start with our new topic, let's watch this video first. Watch and understand why this happened to Ali and how it can be cured. Happy watching everyone! This is Ali. Ali is just an ordinary guy like anyone else. He practices meditation daily and do exercise regularly. He also has a dream on becoming an athlete someday. But there's a major issue stopping him from attaining this goal, and that's rare case of hemophilia. He bleeds a lot from everywhere. He experiences swelling in his joints and there is more blood to lose when he goes to the bathroom. Let's see what causes this problem. DNA molecules are responsible in giving us our characteristics. There are genes in our DNA that synthesize proteins called clotting factors. The clotting factors are what help us clot blood and stop wounds from bleeding. Unfortunately, the gene for a certain type of clotting factor for Ali called factor 7 is not working properly. Now, let's see the effect of this within the body. Here, we have blood vessel and of course it has plasma and red blood cells inside. The blood is flowing normally. Let's say you get a wound. Usually, the clotting factor will stop the blood by sealing the tear. But for Ali, because the gene isn't working properly, so he loses a lot of blood. Can we fix this? Yes. Thanks to recent research, we can fix this by applying recombinant DNA technology, which refers to series of procedures used to join together DNA segments from different species. Now, Ali can be cured. Yes, this time we're going to learn on Chapter 8.0, Recombinant DNA Technology. This technology has allowed scientists to combine DNA molecules from different organisms to create useful gene products. Just look at the visuals here. These colorful fish, monkey, and other living organisms are not the same with the animal we usually see, and this is due to recombinant DNA technology. Alright everyone. Basically, there are three subtopics that we need to cover in this chapter. 8.1 is on the recombinant DNA technology, 8.2 on the method in gene technology, and 8.3 is on the applications of recombinant. So, are you ready to master all these three subtopics? Let's go! Okay, let's start with the first subtopic, shall we? For today's lesson, these are the learning outcomes that you should be achieved. Firstly, you should be able to define recombinant DNA technology. Secondly, define and explain the tools used in recombinant DNA technology. That is the target DNA, restriction enzymes, DNA cloning vector, host cell and modifying enzymes. After that, you should be able to explain restriction enzyme and examples of enzymes that produce sticky ends, such as the EcoR1 at the restriction site GAATTC and blunt end by the smart one at the restriction site CCC and GGG. After that, you should be able to explain the characteristics of plasmid as cloning and expression vector. Then, you should be able to explain the characteristic of E. coli as host cell and its characteristics. And last one, you should be able to explain modifying enzyme and its function. So we're going to focus on the DNA ligase for the DNA ligation and also Taq polymerase for DNA amplification using PCR. PCR stands for polymerase chain reaction. This is the definition of recombinant DNA technology. Formation of new combinations of genes by isolating genes from the organism and then introducing them into either a similar or unrelated organism. This visual explains how the human gene is isolated and is inserted into an egg from a goat. The gene in both organisms is formed and microinjected into other similar or unrelated organisms. Okay, what are the purposes of this technology? So the first purpose is to modify the DNA of an organism to produce new genes with new traits. Secondly, the purpose is to enable scientists to obtain many copies of specific DNA segment for the purpose of studying it. In order to form the combination of genes, we need tools. The tools used in cloning are: the first one, target DNA. Second, we need the restriction enzyme. Third, we need the DNA cloning vector. Four, we need the host cell. And last one, we need the modifying enzymes. In order to modify the DNA or genotypes of an organism, we need to select good genes. This genes is known as gene of interest. The genes are in the selected DNA in a cell. Secondly, restriction enzymes. Restriction enzyme is a molecular scissors that cut or cleave the single strand and the double strand at specific point. Specific point, or the other name for restriction site, is a specific sequence of bases at DNA. For record, restriction enzyme is also known as restriction endonucleases, and it is extracted from bacteria such as EcoR1 extracted from the E. coli, BAMH1 extracted from Bacillus amyloliquefaciens, SMA1 extracted from Serratia marcescens. Most restriction sites are palindromes, that is, symmetrical. What is palindromes? Palindrome is the same 5' to 3' sequence of 4 to 8 nucleotide bases is found on both strands, thus running in opposite directions or anti-parallel, just like the visual given here. These are the restriction sites at the nucleotide bases that enable enzymes such as EcoR1, BAMH1 and SMA1 to act on. Okay, from the visual given here, the enzyme EcoR1 will act at the restriction site 5' GAATTC 3 to 3' and from 5' GAATTC to 3', and so are the others. When restriction enzymes splice or cut at the restriction site, it produces different forms of strand. For example, EcoR1 cut the phosphodiester bond between the complementary bases at A to T, A to T, T to A, T to A, and this producing fragment with sticky ends. Most of the EcoR1 staggered cut into 2 strands forming sticky end, just like the picture given below. Another restriction enzyme is SMA1. It cleaves the center of the bases CCC and GGG, producing fragments with blunt ends. Next tool used in gene cloning is DNA cloning vector. DNA cloning vector is a carrier needed to carry DNA into host cell. These carriers are called vectors. These vehicles are those DNA that can carry a foreign fragment that is inserted into it. This vector can be grouped into different categories such as plasmid, bacteriophages, Cosmin and yeast artificial chromosomes, or YAC. A bacterial cell has only one chromosome, which is made up of a circular DNA molecule, One example of a plasmid is PUC18, which is found in E. coli. This table shows the types of DNA cloning vector and its carrying DNA capacity, the form of vector and also the examples according to its category. We have plasmid, bacteriophage, Cosmin and YAC. To choose cloning vector, we need to understand its characteristics. Basically, a vector must have the following characteristics. Firstly, it must have a multiple cloning site or MCS with a recognition sequence for an endonuclease so that it can be cut to produce a linear plasmid and then incorporated with the new DNA. Secondly, it must be able to accept foreign DNA in multiple cloning sites. The red arrow is showing the MCS. Thirdly, have an origin of replication initiation, that is origin, so that it is able to replicate independently and freely inside the host cell. And the last one, cloning vector also need to possess a selectable marker, a gene resistant to a specific antibiotic. For example, AMPR, resistance to ampicillin. Other than that, unselectable marker that is not less important is LAC-Z gene that encodes for beta-galactosidase. One category of cloning vector is plasmid. Plasmid is not part of chromosome in a cell. It is small ring shape of DNA in bacteria and it has small number of genes. Nevertheless, it has its own origin of replication for self-replicating, and the example of plasmid is PUC18. Another category of cloning vector is bacteriophages. This is a virus that infects bacteria. It can carry larger DNA inserts than bacterial plasmid. For example, is the Lambda 2001. Cosmin is another category of cloning vectors. Cosmin is a hybrid of plasmid and bacteriophage lambda DNA. It can accommodate large inserts of DNA. For example, SCOS1. Bacteria plasmid will not replicate in yeast cells because prokaryotic and eukaryotic DNA sequences use different origins of replication. As such, they are not the good vectors for yeast hosts. Therefore, scientists created artificial DNA molecule called yeast artificial chromosome or YAC to overcome this problem. It has large DNA inserts that can be introduced into yeast cell. The last tools used in gene cloning is the modifying enzyme. These enzymes used to join targeted DNA fragment with the vector to form recombinant DNA. Secondly, this enzyme is used to catalyze the formation of phosphodiester bonds between adjacent nucleotides in DNA. Usually, modifying enzyme that we use in gene cloning is DNA ligase and also Taq polymerase. Host cell is a cell that receives recombinant DNA for cloning purpose through transformation. Host cell is also a cell that can be utilized for DNA cloning in order to accept, maintain, and allow the reproduction of cloning vector. Mammalian tissues, yeast cells, plant cells, and insect cells can also be used as hosts. We also need to select the characteristics in the host cell. Firstly, the host cell should be able to receive DNA recombinant through the transformation process. Secondly, the host cell should be able to maintain the structure of DNA recombinant from one generation to another one generation. Okay, and the last one for the characteristics of the host cell is that it should be able to amplify the gene product from the DNA recombinant. Okay guys, that should cover 8.1. Easy, right? All you have to know is the definition and explain on the tool used in the gene cloning. Now, let's answer these pop-up questions just to check up your understanding in 8.1, okay? Question number 1. A large number of copies of any DNA segment can be obtained by A, B, C, D, or E. Choose the answer. Yes, you are correct. The answer is to introducing foreign DNA into microorganism so that it can be replicated. You are correct. 1 point for you. Question number 2. What purpose do restriction enzymes play in bacterial cells? A, B, C, D, or E? Yes, you are right. B is the answer. Other than to cleave the DNA into fragments, restriction enzymes also attack bacteriophage DNA when it enters the cell. This is for the defense. Please fill up the space given below. Question number 3. DNA ligase links to what DNA fragments by what bonds? A, B, C, D, or E? Yes, the answer should be D. DNA ligase links two linear DNA fragments by covalent bonds. Question number 4. It's about the sticky ends. Remember the sticky ends? So sticky ends are A, B, C, D, or E? Choose the answer. Yes, definitely A is the answer. So sticky ends are the single-stranded ends of a DNA segments that pair with the complementary single-stranded ends. That should be the answer. The last question is, a genomic library is a collection of either A, B, C, D, or E. You need to choose only one answer. Yes, you are definitely correct about this. A genomic library is a collection of DNA fragments that are spliced into plasmids. Thanks for the learning, everyone. This is my YouTube channel. Feel free to visit Roselle Edu TV and watch many more videos on biology. Please like and subscribe. Yeah, you can also scan this QR code to watch a video on the analogy of recombinant DNA. I'm Dr. Roselle signing off for this time. Bye-bye.